Self-contracting muscle implants in mice deliver exercise-like anti-aging effects without movement. This is Planetterrian Daily, episode 166. Researchers implanted self-contracting muscle constructs into older mice and observed reductions in age-related decline markers, comparable to those seen with voluntary wheel running. The implants generate repeated mechanical contractions that activate pathways tied to mitochondrial biogenesis and reduced inflammation. The constructs were designed to mimic the rhythmic activity of skeletal muscle during physical activity. Follow-up experiments will test whether similar constructs improve outcomes in models of sarcopenia and metabolic disease. This approach isolates the contribution of mechanical force from other exercise-related factors, such as increased blood flow or hormonal changes. The work highlights how muscle-derived signals alone can influence systemic aging processes. Most people assume the main benefit of exercise comes from burning calories or building visible strength. In reality, the repeated mechanical contractions of muscle tissue itself send signals that affect mitochondria, inflammation, and metabolic health throughout the body. Right now, even while you are sitting still, your existing muscle fibers maintain baseline tension that supports these pathways at low levels. Skeletal muscle accounts for roughly 40% of total body mass in adults and is a major source of circulating myokines. When contractions are absent for long periods, those signals drop and age-related changes accelerate in multiple organs. The recent mouse implant study shows that artificial contractions can partially restore the same signals. A practical takeaway is to look for any safe way to introduce regular, low-level muscle activity, standing, stretching, or resistance bands, rather than focusing only on intense workouts. From artificial muscle signals to another way of correcting faulty cellular instructions, we turn to a genetic approach using tRNA. A new delivery method using transfer RNA rather than messenger RNA successfully rescued nonsense mutations in cystic fibrosis cell models. University of Toronto researchers demonstrated restored protein function in patient-derived cells. The technique allows read-through of premature stop codons without triggering strong immune responses seen in some messenger RNA platforms. The method exploits the natural role of transfer RNA in translation to bypass faulty stop signals. This avoids the need for full gene replacement or permanent DNA editing. Next steps include testing in additional genetic disorders caused by nonsense mutations. The University of Toronto transfer RNA delivery work opens a route for treating genetic diseases caused by nonsense mutations that current therapies cannot address. It could eventually help patients with cystic fibrosis and dozens of other rare conditions where premature stop codons truncate essential proteins. Watch for animal studies that test delivery efficiency and off-target effects across different tissues. While transfer RNA offers a new genetic tool, another study examines how suppressing cancer genes can sometimes allow cells to survive in a senescent state. A recent study tested whether blocking key oncogenic signals leaves some cancer cells able to enter a senescent state and later reactivate. In cell models, temporary oncogene withdrawal permitted a subset of cells to persist rather than die. Researchers used timed drug exposure to mimic clinical treatment schedules. The work suggests senescence may act as a temporary refuge that later fuels relapse. Longer-term experiments will examine combination approaches that target both proliferating and senescent populations. The findings challenge the assumption that oncogene inhibition alone is sufficient for durable tumor control. Senescence and health span also appear in a new framework that links geroscience to daily behaviors. The proposed set of 8 evidence-based factors integrates geroscience findings with everyday health behaviors. It expands on cardiovascular metrics to include sleep, physical activity, and social engagement as measurable contributors to health span. The framework draws on longitudinal cohort data linking each factor to mortality and morbidity outcomes. The article outlines how clinicians could track these factors alongside traditional biomarkers. Implementation studies are planned to test uptake in primary care settings. It aims to translate cellular aging research into actionable public health guidance. From broad behavioral frameworks to a hands-on citizen project measuring soil biology, we turn next to microbial activity in the ground. 1,000 volunteers buried organic cotton underwear across Switzerland to measure how the land use affects microbial breakdown. Agricultural soils decompose the fabric faster than forest soils, pointing to differences in microbial communities and moisture. The project standardized burial depth and fabric type to enable direct comparisons. Decomposition speed varied markedly with soil pH and organic matter content. The simple, low-cost method provides a standardized proxy for soil biological activity. Results will help refine models of carbon cycling in different ecosystems. Soil and environmental factors also shape human risk, as shown in a study of landslide exposure. A study mapping landslide risk across Appalachia shows that residents with fewer resources face higher exposure because they live on steeper slopes and have limited ability to relocate or reinforce homes. Wealthier urban areas can invest in drainage and retaining structures that reduce incidents. The research combined geological data with socioeconomic indicators to identify hotspots. The analysis revealed clear disparities in both hazard exposure and mitigation capacity. Communities with lower median incomes showed consistently higher vulnerability scores. Improved early warning systems and targeted infrastructure support are proposed as immediate next actions. Another environmental signal, fluid injection, proves hard to predict from small events alone. Analysis of 70,000 earthquakes linked to fluid injection found that foreshocks precede only a minority of larger events. The study examined cases where operators could have paused injection based on early signals, but larger quakes still occurred. The dataset spanned multiple injection sites and time periods. Researchers note that fluid pressure changes can trigger events without clear seismic precursors. The findings suggest current monitoring thresholds may miss some high-risk sequences. It underscores the difficulty of relying solely on microseismic activity for operational decisions. Finally, institutional news from a Canadian university. The university named 3 scientists to Eddie Goldenberg research chairs focused on areas including regenerative approaches and chronic disease mechanisms. The positions provide sustained funding to accelerate translation of basic findings into clinical applications. Each chairholder brings established programs in cellular and molecular biology. The appointments expand the university's capacity in fields that intersect with aging and tissue repair. Recipients were selected through an internal competition emphasizing translational potential. The chairs are named after a donor whose support targets high-impact biomedical research. Next time, keep an eye on follow-up animal testing of the transfer RNA platform for other nonsense mutation disorders. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NARA Network, links in the show notes. This show comes to you from the NARA Network. If you want another sharp daily briefing, Offshore North has you covered. Offshore ocean racing explained every Monday with a Canadian angle. All our shows are free at naranetwork.com. Live dashboards, TSLA, SpaceX launches, the investing scoreboard are all at naranetwork.com data. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.